Hello, and thank you for coming today to Grand Rounds. Welcome. This is Grand Rounds for the Minnesota Regional Spinal Cord Injury Model Systems, or we call it MINSCIMS for short. And thank you for joining us today. So MINSCIMS is funded by the National Institute on Disability, Independent Living, and Rehabilitation Research, NIDILRR, Spinal Cord Injury Model Systems Program. And the purpose of the SCIMS is to support innovative projects and research in the delivery, demonstration, and evaluation of medical, rehabilitation, vocational, and other services to meet the needs of individuals with spinal cord injury. MINSCIMS represents an exciting new partnership among the following institutions: Courage Kenny Rehab Institute in Minneapolis, Minnesota, Mayo Clinic in Rochester, Minnesota, Regions Hospital in St. Paul, Minnesota, and the University of Minnesota in Minneapolis, Minnesota. Our community collaborators include Minnesota Spinal Cord Injury Association, Unite to Fight Paralysis, Get Up, Stand Up to Cure Paralysis, Paralyzed Veterans of America, United Spinal Association, and the North American Spinal Cord Injury Consortium. This system collectively provides a multidisciplinary continuum of care for people with spinal cord injury, including emergency medical services, acute care services, acute rehab services, and post-acute services. My name is Dr. Angela Hengst-Philippus, and I will be the moderator for today's session. I'm a research professional in the Department of Rehab Medicine at the University of Minnesota Medical School, and I'm a co-investigator on SCIMS, the MINSCIMS project, that is. All right, a couple of housekeeping notes. So the University of Minnesota has organized the CME for today's webinar. Please see the chat for information on how to record your attendance and claim CME credit for attending this webinar within 24 hours of the event. This meeting is being recorded and will be posted on the MINSCIMS website. And to earn CME credit for today's presentation, please click on the link in the chat. Due to the number of registrants for today's webinar, we've placed phone lines on mute to reduce audio feedback. However, we do welcome and encourage audience participation using the Q&A function. If you have any technical or logistical questions, we ask that you please submit those in the chat to Dan Kelly. We extend a huge thanks to all of those who work behind the scenes to make these webinars possible. The title of today's presentation is Cell Transplantation to Improve Sensory Recovery After Spinal Cord Injury. And the speaker for today's webinar is Dr. Ann Huntemer-Silveira. Dr. Ann Huntemer-Silveira is a postdoctoral associate in the Parr Spinal Cord Injury Lab at the University of Minnesota Stem Cell Institute. Her research focuses on stem cell modeling for spinal cord injury and the development of stem cell-derived spinal interneurons to support sensory recovery and cell transplantation models. This work was funded by the Minnesota Office of Higher Education SCI TBI program, as well as an NIH R01. Today, she'll discuss her research in this area and some of the challenges associated with the study of sensory function after spinal cord injury, both at the bench and in the clinic. So welcome, and thank you so much for presenting.
Thank you very much for the introduction and for the invitation to present to you all today. This constitutes a big chunk of my dissertation work, so I'm very excited to share it with all of you today. Dr. Ann Parr, unfortunately, was in clinic today, so she wasn't able to make it, but I have included her contact information at the end of the presentation if anyone is interested in learning more about her clinical work. As already mentioned, portions of this project were funded by the Minnesota Office of Higher Education SCI TBI Research Grant Program, as well as an NIH R01, and I have no conflicts of interest to report. So to start us off with a brief overview of what I'll be talking about today, our focus of discussion is sensory function after SCI. So I'll start with some general background about what sensory function looks like after injury, how therapeutics have generally been used as a treatment for spinal cord injury, and we'll dig in a bit to that question of how and whether cell therapeutics are able to support recovery of sensory function. Then I'll move into some of the projects that we work on in the Parr Spinal Cord Injury Lab, ranging from making cell types that identify those sensory circuits, supporting graft integration in those sensory circuits, and then finally studying sensory circuits in our spinal cord injury models. Since this is a more clinical audience today, I also wanted to take a brief moment to look at the translational research spectrum and sort of show you where on this spectrum our research falls. The Parr Spinal Cord Injury Lab has research that spans all the way from basic science all the way to clinical trials and practice. But our cell transplantation work falls right here in between basic science research and translation to humans, where we are using human cells and GLP compliant materials and reagents. But we do currently work in animal models and are in preparation of moving towards clinical translation. So, sensory function after spinal cord injury. This can refer to both loss of sensation or sensory paralysis, as well as chronic pain. Both are very common after injury. But I do want to note here that loss of sensation or sensory paralysis tends to get wrapped up with motor paralysis, and in animal models, loss of sensation is rarely studied. So for the majority of this presentation, I will be focusing on the pain aspect of sensory function after injury. Within that, it's estimated that as many as 60 to 80% of SCI patients will experience chronic pain at some point after their injury, and roughly a third of these patients classify that pain as severe or debilitating to everyday life. The pain experience after injury is very heterogeneous. It constitutes both nociceptive and neuropathic types of pain. And this has a major impact on quality of life, where we see both individuals with nociceptive and neuropathic pain report substantially lower quality of life indices compared to individuals who report no pain. And really importantly, the impact of pain has largely been considered a priority in the treatment plan for individuals with SCI. And this ranking as a priority tends to increase with time after injury, though generally it is not highly ranked in priority in a lot of research programs. When we look at current approaches in pain management after injury, unfortunately, our best and brightest right now is largely restricted to sort of pain management and reduction of symptoms. This includes pain medication via pill or pump, acupuncture, massage, physical therapy, and exercise. There are also newer treatments related to electrical stimulation. You may have heard of transcutaneous electrical nerve stimulation, TENS, or transcranial electrical stimulation. And then finally, we also have our experimental treatments in the preclinical stage. And it is really here that we have a lot of the hope for being able to develop treatments that can actually reverse the effects of sensory dysfunction after injury rather than just treating symptoms. These include small molecule and drug therapies, as well as cell replacement therapy, which will be the focus of what I'm talking about today. So cell replacement therapy, also referred to as CRT or cell transplantation. I'll use those terms interchangeably. This refers to transplantation of stem cell-derived neural cells into the site of the injury, with the goal here being to replace the tissue that has been lost and damaged and provide cells that can hopefully jumpstart the system into meaningful recovery. In preclinical models, there have been many different applications of cell transplantation, and they have been associated with many different mechanisms of action. These include neuroprotection, remyelination, immunomodulation, axon regeneration, as well as relay formation. However, clinical trials have been a bit more mixed. We have largely established safety of cell transplantation in the clinic. However, functional outcomes have been mixed, and it's been really difficult to elucidate possible mechanisms of action. And there are many reasons why this might be, and many contributing factors. I'm going to focus on those related to the cells themselves and the extent to which these cells are able to integrate with host tissue. By that, I mean the extent to which when we put the cells in, whether they just sort of sit there or whether they're really able to form meaningful connections to and from the host spinal cord. And that is really essential, especially for chronic spinal cord injuries, to be able to get any kind of meaningful functional benefit. So when we go back to the bench and we look at ways that we can really improve graft integration, there are a couple of really important areas to focus on. These include developing protocols that can make these cells quickly and with high replicability and reproducibility, which is, of course, always difficult across many different fields of science. There's also a really big need to improve the extent to which these cells have structural support. And what that means is some kind of support or guidance that can encourage grafts, connections between the graft and host tissue. Most neurons at this point have been shown that if they're just injected into the cavity, they're not going to move across an empty space. So they really need some kind of structural support to encourage them to extend axons both to and from the host spinal cord. And finally, we have regional specification. And what this refers to is patterning of the transplanted cells to the level and phenotype of where the injury is. Intuitively, it makes sense that cells that go in should look like what was there before, but historically, this has not been the case. But increasing research has shown that this may be really, really important in improving this sort of host-to-graft integration. A major reason for why that is so important is because the spinal cord itself and the very anatomy has really distinct functional domains, such that the sensory and motor deficits that we see following spinal cord injury are generally able to correspond to damage in the dorsal and ventral spinal cord, respectively. This is a bit of a simplification, and there's a lot of interconnectedness between these two domains. But for the most part, when we're talking about these discrete functions, we see sensory information coming in from the body through the dorsal spinal cord up to the brain, and motor information coming from the brain down through the spinal cord and out to the body. And when we appreciate how distinct these two regions are of the spinal cord and how precise their functions are, it becomes clear that it will be really important that cells that go into this area are actually able to meaningfully incorporate with these two distinct circuits. With that said, this functional specificity historically has not very well been incorporated into cell transplantation work. What that means is when we're looking at the differentiation protocols that we use to go from these stem cell populations to spinal neurons, this is most commonly done via growth factor-mediated differentiation paradigms. And generally, the goal here is to really recapitulate the relevant stages of neurodevelopment. What this typically looks like, you'll start with a period of neural induction where you're restricting the fate of those stem cells specifically to that of the central nervous system. This is then followed by a period of rostral-caudal patterning, where you're further restricting these cells along the rostral-caudal axis, in this case to a spinal cord identity, and then even further restriction to very specific regional patterning. This is what gets us all the way to those different spinal interneuron populations, motor neurons, and so forth. Now, when we look at the history of cell transplantation literature and what types of cell populations we have been making, the vast majority of protocols that we see carefully follow this neural induction stage, but often don't go too much further. This is where this research really started, and it has worked surprisingly well in animal models, but it is increasingly clear that this level of patterning is not sufficient when we're going all the way up to the human model. A small but growing number of studies do actually take this a step further and go all the way to patterning at the spinal level. Interestingly, most clinical trials actually don't use or haven't used neurons that actually have the spinal identity, but they actually have a brain phenotype. And that is another indication for why we may not be seeing as a strong integration in our human trials as we have in animal models. And then finally, even fewer protocols go all the way to this very specific level of discrete patterning. And when we look within this, we see even more of a gap. So the vast majority of those protocols that are doing this level of region-specific patterning overwhelmingly focus on locomotion and motor recovery. Substantially fewer have spent time creating populations related to pain and sensation. One sort of crude but very effective way to look at this gap in our research is to simply use our databases and our Google searches. So this is a very simple example, but if we pick our favorite database, in this case, let's say PubMed, and we look up spinal cord injury motor, we get about 18,000 results. If we just change motor to sensory, that drops all the way down to 6,000. If we change out sensory for pain, we do get about double the number of responses, but we still have a substantial gap between our sensory search terms and our motor search terms. And this isn't even restricting to specific types of study or article. And this holds up in pick your favorite database. We see it in Web of Science. We even see it in Google and a variety of other databases. This sort of trend in just quantity of research that's been published in this area has a major gap. The question of why this gap exists gets a bit philosophical, and I won't go too much into that at this point. But really the take-home message here is that this gap does exist. And this is doing a disservice to our patient population and the community who have been saying for a while that their pain management is a major component and goal of their treatment plan. To explore a little bit more about why pain and sensory function have historically been so limited, we do need to talk about the fact that it is actually really hard to measure pain and sensory function, both at the bench and at the bedside. So clinicians and researchers alike can go on for days about some of these challenges. To summarize them, when we're talking about humans and looking at and measuring pain, these metrics are largely dependent on self-report, which are not always the most reliable. They are, of course, subjective, and they do vary by individual tolerance. So what is really overwhelming for one individual may be barely registered by another. And even more frustratingly, especially for patients and clinicians, is that the cause of chronic pain is often unknown in these individuals. The story does not get easier in animal models. So we can't ask our animals how their pain is on a given day. So this means we have to measure it somewhat indirectly. And this is primarily done through the use of behavioral measures. And the gold standard for this really requires an intact withdrawal response. So for example, in a rodent model, you may be stimulating any of the fore or hind paws and looking for a withdrawal response and measuring how that withdrawal response changes over time under different conditions. This is, of course, a major issue for spinal cord injury, where our injury models themselves impair the ability of these animals to have a motor response. So this introduces a number of difficulties and confounds to this work within injury models. And there are indirect measures that can be used to look at pain-related behaviors. These are often related to stress behaviors, such as anxiety, depression, aggression, and so on. But of course, both humans and animals following spinal cord injury do have a substantial increase in stress-related behaviors, which may or may not related to a current pain state. So this also doesn't really give us a good understanding of what's going on in terms of the sensory function of these animals after injury. And again, even in our animal models, there's great variability and individual tolerance and pain response in these animals. So all of this is to say that measuring pain in our animal models and in humans is very difficult. There are a lot of different factors that can confound this research, and it makes the data really messy and often really difficult to publish. This does not mean that we shouldn't be doing it, but it means that we do need to do a lot more work to get clear and robust measures. So if we take all of this together now, and we ask the question of what's going on with cell transplantation and using it to study sensory function, our first question that has really come up and posed a major problem in this field is whether cell transplantation can inadvertently actually cause chronic pain. The answer to that is yes. There have been studies that have been published using cell transplantation, and that rather than improving a sensory state, actually contribute to a chronic pain state. Multiple studies have found this. And what I want to point out here is that both of the studies featured here, as well as most of the others that have utilized cell transplantation looking at induction of chronic pain. They use cells that are non-specified. So they have a general neural identity, but they have not been patterned specifically to the sensory system. And also really importantly, when these grafts were looked at after transplantation, rather than these grafts becoming neural cells, they in fact often become astrocytes, which astrocytes also have a long and complicated history with chronic pain, both in injury and in other disease models. So the picture is complicated. And then we also need to ask the question of whether cell transplantation is able to improve chronic pain. And the answer to that question is also yes. If we incorporate regional specification to really be able to control the final identity of our cells when they are transplanted, and often if we are able to incorporate a combinatorial measure, such as some kind of training, to really be able to guide and encourage those cells in their development, this has been shown to really substantially enhance both integration and functional improvement after transplantation, and decreases the risk of that maladaptive plasticity that other papers have shown. This is one such paper featured here. And another component of this work is that when we do look at, as I mentioned before, the much smaller proportion of studies that incorporate this regional specification, while we do have dozens of protocols that have generated all of the different ventral interneuron populations and a variety of motor neuron populations, to date, only a few have produced some of the dorsal neuron populations. Even fewer of those protocols have been used in SCI, and even fewer still have been used to study sensory function after SCI. So there is a major gap here that really needs to be addressed and filled. And this brings me to the main focus of the research and the work in the Parr Spinal Cord Injury Lab, and our major goal here, which is to build a pipeline that can really better support sensory function in cell therapeutics. So there are 3 different buckets of research that I'm going to walk us through today. The first is starting with just making these regionally specified sensory populations and what they look like. The second will be really creating an environment with those cells that's able to better support graft integration. And then finally, we will talk about actually studying the changes in sensory function in our animal models. So starting with making the cells, the abbreviation that I'll be using throughout here is spinal neural progenitor cells, or SNPCs. And in our lab, we use a human-induced pluripotent stem cell model. So we start with these undifferentiated HIPSCs at day 0. And we have a patented 24-hour neural induction approach where following 24 hours, we have a primitive neural ectoderm population. This population can be further cautelized to that spinal identity. And then from there, we can split this population up to produce both dorsal and ventral progenitor cells from a shared parent lineage. We get these progenitors at about 6 days of differentiation, and then we can mature them for a given period of time. We do see that both dorsal and ventral populations achieve a largely postmitotic identity by about day 20 in culture. So I'll focus for the rest of this section on these dorsalized sensory populations. And when we look a little bit more closely at their characterization and what constitutes what can be considered a dorsalized neuron, we have our main stages from that early induction period all the way to when they reach that postmitotic state at around day 20. When they get to that progenitor stage, this is really characterized by robust expression of dorsal progenitor markers and a lack of expression of similarly robust ventral markers that are expressed in our ventral populations. This follows all the way through to that postmitotic stage where we switch from progenitor markers to postmitotic interneuron markers. And when we look at these cells a bit more broadly, we see that over time we do switch from day 2 to 20 to a high proliferation marked by Ki67 staining to non-proliferative and postmitotic. And in terms of the distribution of the neural identity of these characters, there's some variability across cell lines, but we do see about 60 to 80% neuronal, and most of the remaining cells are astrocyte in origin. So similar characterization has been done in our ventral populations, but I won't focus on that right now. But now that we have these two different populations, we are able to play with them in all kinds of fun ways. I do want to point out that these iPSC-derived SMPCs do meet our clinical GLP requirements for translation, and that is an ongoing focus of the work in our lab. As I already mentioned, this meets the requirements for being a fast and reproducible protocol. Being able to make post-mitotic neurons in 20 days is among the fastest protocols out there, and we are able to incorporate that regional specification that has been shown to improve graft integration. We are studying these two different populations, both in isolation and in co-culture, to really be able to determine their functional profile on their own and in more complex cellular conditions. And we're doing this across multiple different cell lines. We are currently using a clinical grade HIPSC cell line that is in current clinical trial for spinal cord injury in Japan, which is also a really exciting avenue of research for us. Finally, of course, our ultimate goal is to use these cells in combination to really be able to observe how we're able to contribute to greater graft integration to recreate these proper spinal cord circuits. Both of which, these in vitro models and these in vivo models, we believe are really important to advance our cell therapeutic strategies from bench to bedside. So that's the production of these cells. Moving now to creating an environment that supports transplantation of these cells. What we've really found is that 3D culture is a really essential system in order to be able to produce cells that are capable of these complex interactions. It may not be surprising, and at this point it is well published, that cells that are grown in the standard 2D system, such as what I've already shared with you so far, are generally just not equipped for that complex 3D environment. You can see in the schematic here on the right, there are really profound differences in this environment where going from 2D to 3D, we have substantial differences in cellular polarity, access to nutrients, the provision of extracellular matrix support. All of these further influence the way these cells are able to adhere, extend, and migrate. And this also ultimately influences the way these cells are able to differentiate and form functional connections. Because of this, it comes as no surprise that cell fate in these 2D monolators very rarely matches what happens when those cells are transplanted in vivo. So given this information, we made the decision to move our cultures away from these more standard 2D models into a 3D culture system. And we do this using a scaffolding system that we found really guides cell-to-cell contact. So what this means is we have 3D printed support structures, which we'll refer to as scaffolds. And in our work, we've shown that these scaffolds are able to really support the formation of three-dimensional cellular interactions. And they are very effective at guiding growth both into and out of the transplants. This work was done in collaboration with the Dr. Michael McAlpine's engineering lab at the University of Minnesota. And what I've featured here in our scaffolds, the ones that I'll be talking about today are made from silicone, but we have tested other materials as well. And these silicone scaffolds are 3D printed to contain these 3 microchannels into which our cells are directly 3D printed into. What that looks like here, you can see a close-up of one of those scaffolds, and the light pink is our BioInk, which contains a mix of Matrigel and our spinal neural progenitor cells. So we print these cells and then we grow them for about 3 weeks in culture to allow them to form those dense 3D networks, which we do see what we refer to as an assembloid here, where we have very strong expression of neural tubulin markers, creating these dense 3D neural structures. So with this 3D model, we can then better characterize how this model might be different from our standard the more standard 2-dimensional differentiation and maturation approach. And we do actually see very substantial differences between our cells when they are cultured in 2D versus when they are cultured in 3D. Looking specifically at the identity of that dorsal sensory population, we do see here that when we culture our cells in 3D, we get enhanced maturity. Our 3D assembloids do in fact exit a proliferative state much faster, and they also exit the neural progenitor state faster, indicated by markers of Ki67, Nestin, and PAX3, where generally speaking, these cells seem to be maturing at a more rapid rate. We also see differences in the ultimate differentiation of our cells in 3D as compared to 2D. As I already mentioned, our 2D cultures produce about 60 to 80% neurons. And in our 3D cultures, this is actually shifted to more of a 70-60% neuron and 30-40% astrocyte. And given that we know that cells, when they are transplanted in vivo, they very often produce a much lower percentage of neuronal populations, we do believe this to be in line with what we would expect in an in vivo model. We also see these cells expressed a higher proportion of mature adult interneuron markers shown in our histology in 2D and 3D, as well as in qPCR data shown at the bottom. And in general, we see a wider array of synaptic and mature functional markers as well. So all of this has been focusing on our dorsal sensory populations. We have also done similar characterization as well as sequencing in our ventral populations. I won't talk about that much today, but that publication just came out this year for anybody who is interested. And in general, these findings are consistent across our different populations. We do see very distinct clustering differences between our 3D populations and our 2D populations. When we look at electrophysiology, our 3D assembloids do have much more diverse functional activity. And we also see increased expression across the board of many different markers associated with neurodevelopment and regionalization in our 3D as compared to our 2D cultures. What all of this means and how this has implications for cell transplantation is that we see with our system that generally our more simple and traditional culturing methods are not sufficient for robust cell transplantation outcomes. This includes adherent monolayers in our 2D cultures, and as also direct injection, where we're not providing any sort of structural support to ensure that the cells are able to grow into and out of the grafts. We have also found that by increasing the complexity of our culture system and also using them in combination with other approaches, this really can provide the best outcomes both in preclinical and clinical transplantation models. In our systems, we use 3D culture and physical scaffolding. There are a myriad of other scaffolding approaches that have been done that have been shown to have very beneficial effects, as well as neurotrophic support as well. So there are a lot of options out there for mixing and matching to produce a robust cell transplantation approach. So with all of that in mind, we're going to move to the really exciting part now, which is our animal models. So the studies that I'm going to be sharing with you today are specifically using these 3D scaffolds. This data uses an acute transection model. And we have done these studies both with our dorsal populations and our ventral populations in isolation. And I'll be sharing bits and pieces of both. So the timeline here for our in vivo studies, we begin with differentiation of our HIPSCs, and we do differentiate ourselves for the first 6 days in that standard 2D monolayer. At day 6, when our cells have reached that progenitor identity, we take them and we 3D print them into our scaffolds, and then we allow them to develop into those complex 3D structures for about 3 weeks. At that 3-week time point, we transplant them into our SCI model. We use an apyemic nude rat to account for the xenotransplantation. And we also use a mid-thoracic injury, so that's the T8 complete transection. At this point, our animals undergo weekly sensory motor testing, and at 3 months post-injury, we look at downstream neuroanatomical effects. I do want to point out again, also, in this case, we are transplanting the scaffold and the cells, and this does require a transection model, but we are also currently working on adapting this to a contusion model, where we are actually able to create a release system with our scaffold so that we can remove the assembloid from the scaffold and transplant it in a scaffold-free system, specifically for contusion models. So that is really exciting ongoing work to be discussed further at another time. As I said, we have transplanted this with our dorsal cells and our ventral cells. I'm going to look first at the pilot data that we have transplanting our sensory populations. This is just a pilot study, and it was just done, just finished this year. So this data is still really preliminary and is still actively being analyzed. But just to give a few of the highlights, what we see when we look at these cells anatomically, Shown here on the left, we have the spinal cord with NF200 positive neurofilaments, and in green, we have our transplanted cells. This particular section really nicely shows the anatomy, where you can see our 3 channels of that scaffold. At this particular angle, we're mostly able to see the cells on the lower half, and they do, in fact, fill the channels of the scaffold and also migrate out of the bottom and the top, but you can't see that in this image, above and below the site of injury, and do incorporate with host cells. When we look more closely at these cells, we see widespread expression of neurofilament markers. And when we analyze the profile of these cells, we do actually see that they maintain a consistent percent positive neuronal population with what we assessed in vitro. So we see about 60 to 70% MAP2 positive neurons and about 30 to 40% S100 positive astrocytes. So it's really exciting that this work was consistent from our in vitro data to our in vivo data. We do see also positive expression of markers for regenerating axon, as well as synaptic proteins that are observed within contact of our grafted cells and our host to graft connections. There we go. So all of this gives us preliminary evidence that these cells are both migrating into and out of the host spinal cord and potentially forming meaningful connections here. So from a functional standpoint, this data is really exciting. We have seen very similar work with our ventral transplant study, and that study, as I mentioned before, was published earlier this year for those who want to see that data. Shifting then to the functional recovery, there are a lot of different ways to measure functional outcomes. Again, we're looking primarily at the hind paw because this is a thoracic level injury. Looking at motor function, we use the BBB locomotor scoring, which is fairly standard for rodent transplantation studies. For sensory function, we look at both light touch and punctate pain using a cotton swab test, which is like a light foot tickle, and von Frey withdrawal filaments, which allows us to evaluate punctate pain. Now for the ventral transplantation, I won't show that data for the time being, for the sake of time. We do see substantial motor function and recovery in that model. But at the time that that study was done, we were not actively doing sensory function except to assess whether or not these animals developed chronic pain, and we didn't see that there. With regards to our dorsal sensory transplant study, I'm so sorry, but I have done a bit of a bait and switch here. So as I mentioned before, we've only done a pilot study so far with not nearly enough animals to be able to clearly assess behavioral outcomes. So stay tuned here as we continue to run more animals and expand our data set. I can't make any conclusive statements, but there are some early trends that I do want to share that are giving us a lot of really valuable information for better understanding what sensory function looks like in these injury models. The first thing is that what we're really seeing here is that sensory recovery, to the extent that we're able to measure it, really occurs at a much slower rate than what we see for motor recovery, which has been fairly well documented in rodent models. And this is especially true in a severe injury model. With what we've seen so far, we have not seen any evidence that animals that do receive these dorsal scaffolds develop chronic pain. So that seems to be not a concern that we have at this time. But when we look at our von Frey filaments, what we see here in a very small percentage of animals shown over time, we're increasing the strength of that von Frey filament, with an 8 being the highest stimulus that the hind paw is receiving. And we don't see any differences between groups at this time, but what we do see is that while with motor recovery, it's generally accepted that most animals will show endogenous motor recovery within the first 3 weeks, and any improvement thereafter is considered related to the treatment. That is not the case for sensory recovery in these animals, especially in a severe transection model. We do not begin to see any changes in endogenous sensory state until around week 6 or week 7. And what you'll notice, especially in these thicker filaments, is that at week 12, at our final time point, animals across all groups are still recovering. So what this indicates to us is that the timeline of sensory recovery, to the extent that we're able to actually detect it in these animals, is still ongoing and occurs at a much slower rate. So we may need to look at longer periods and able to actually get to the point where sensory function is able to plateau, and we're able to really look at the differences between our control and our treatment groups. We did also get a bit of a surprise here, where we're seeing basically preliminary evidence of a forepaw effect, which has not been documented well in the literature and was not expected for a thoracic level injury. Again, very few animals currently to report in here, so the N is small. But we do see this trend, and it is consistent across all of the different sensory tests that we've done. So we suspect there may be something here to investigate further. In this case, we stimulated the left forepaw as a control because the forepaws are not expected to be implicated in a thoracic level injury. Go figure. It seems that in our model, the forepaw may be in fact implicated in injury. And what we're seeing here is that our control animals, both the animals that received only the injury or an injury with an empty scaffold, actually have decreased sensitivity in that forepaw, and that decrease in sensitivity persists throughout the course of the study. That decrease is not evident in the animals that received the cell scaffolds, and we suspect this may be related to some sort of neuroprotective effect, but that histology analysis is still ongoing. So I don't have an answer to this question yet, but it is a really interesting finding that we found, and there's not a lot on upper level effects of injury in these rodent models. So if anybody knows more, I'd love to hear about it. And for the most part, that is a wrap. So to go into details on our future work, sensory function in general really remains poorly understood. Better systems are in progress. We're working on this question as well as several others. And the questions that we really want to look into moving forward include how sensory recovery really changes, both across different injury models as well as different levels of severity. And we also want to further study what really contributes to the development of chronic pain after injury and or in our transplantation models. Research also typically reduces the scope to individual cell types and systems. The data that I have presented to you today has mostly been of our dorsal or ventral cells in isolation. But another major goal of our research is to combine those systems together, because translation from animal to human models really requires a more complex system that incorporates all of the different spinal domains. So our major ongoing work is modifying our 3D pipeline to really be able to combine both populations into biologically relevant arrangements to create a full spinal assembloid. And with that, I can take questions. I do have our contact info at the top, my email and Dr. Parr's for anyone interested in that clinical work that I wasn't able to go into today.
Excellent. Thank you so much for a really awesome presentation. So like Dr. Huntemer-Silveira said, she will take your questions now. So feel free to type any questions you have into the Q&A box, and I can read those out loud. Not seeing any questions come through at the moment. People might still be typing. How much time do you have left on your R01?
This is the last year.
Last year. Okay, very
And of course,
exciting.
being up for funding right now is a bit nerve-wracking, but things are looking up. We've got a lot of solid work coming up for the future. And I know a lot of the data that I presented today is really preliminary. So there will be more on a lot of the projects that I've shared today moving forward as well.
That's great. And maybe we can have you back to talk about those in the future, progress of those.
Happy to come back.
Yeah. Well, I'm not seeing anything come through right now. That was a lot of great information people might need to digest, and they can always, if you want to put up your final screen again, people can reach out to you directly if they have other questions. There we
Here
go.
is contact information. I can also drop that briefly in the chat.
Yeah, that would be great. Wonderful. All right, now we can probably go to that last slide then. Right. So thank you all for attending today's grand rounds. And we apologize if your question wasn't answered or if you didn't have enough time to type it in. Feel free to reach out to our presenter directly if you would like to follow up regarding your question. So the recording again for this webinar will be posted very soon on the SCIMS website with a link to our YouTube channel for you to view it. As a reminder, please see the chat information on how to obtain CME credits for attending today's webinar. And then at the end of the webinar, a brief 3-question survey will pop up, so we would appreciate your feedback. Thank you all for coming and have a great rest of your day. Thanks, Anne.